In this video, I will demonstrate how to do the dilution series, and then how to spread those liquid culture suspensions of yeast with a hockey stick spreader onto a plate. This is a different technique from streaking, which I demoed in a different video. For our yeast mutagenesis experiment, we want yeast cells evenly spread out on the Petri dish surface, and ideally, once those cells grow up into colonies, we don't want the yeast colonies touching each other. So we want as many colonies as we possibly can have while also giving them room, and so without them touching. That is approximately about 200 colonies. In truth, if we were to do a formal mutagenesis experiment, you would actually want to officially count the yeast cells in your culture before plating them onto a plate. That way you officially know how many cells are on that plate. For us, however, we will approximate the number of cells that we are plating, and that is why we are doing this dilution series. So we are doing it the quick and dirty way. It isn't so important for us to be completely accurate. First thing is to set up your test tubes. You will have a rack like this with 5 sterile test tubes. You will be working with your lab partner on this. Go ahead and label the tubes 10 to the 7, 10 to the 6, 10 to the 5, 10 to the 4, and 10 to the 3. In the 10 to the 7 tube, you will put 1 ml or 1,000 microliters of sterile water, and the others only put in 900 microliters. You will have a handy device like this called a pipette, and this is actually a micropipette, so it will be able to transfer very small volumes of liquids such as 1 milliliter or less, and you also have a P200. And FYI, for those of you using this for the first time, there are 2 stops on the plunger. There is the first stop that you will feel when you press down, but then you actually can press down a bit more to feel the second stop. Do not go down to the second stop before pipetting up liquid. Go to the first stop, then slowly release to pipette up liquid. Then when you go to expel the liquid, you can then go down all the way to the second stop to completely expel everything out. The reason for this, when you go all the way down to the second stop, you are actually expelling more than the set volume. So if I were to go all the way down to the second stop and then slowly release the plunger, I would actually have more than that set amount of liquid. So don't do that. But do go all the way down to the second stop when you are expelling. Here's my P1000. I'm putting on a fresh tip. I will now set it to 1,000 microliters. This is what it will look like. That's 1,000. I will now pipette up 1,000 microliters of water. So you can see I have water in the tip. I am now going to take my 10 to the 7 tube and expel that 1,000 microliters into that tube, making sure that I close the lid to that tube. Eject the tip. Now I'm going to set it to 900 microliters. New tip, and the rest of the tubes get 900 microliters. So 10 to the 7 is our largest number here, so that is going to have the most yeast cells. And then what we're going to do is a 10-fold dilution of this to concoct the 10 to the 6 suspension of yeast. And we'll do that by taking 100 microliters of the 10 to the 7 and diluting it up to 1 milliliter or 1,000 microliters. So for that, we will need 900 additional microliters of water that we're adding to that 100 microliters that we took out of the 10 to the 7. And thus we are diluting it 10-fold. We will then dilute this 10 to the 6 10-fold to make the 10 to the 5. By doing the same thing, taking 100 microliters of the 10 to the 6, diluting that up to 1 ml by adding it to this 900 microliters of water here, and so on until we get to the 10 to the 3, which will then have the least number of yeast cells in it. It will be our lowest concentration of yeast. So now I will prepare the 10 to the 7 mixture. What you will do is get a toothpick full of HBO yeast. This time you do want to see a small glob. When you do the streaking and setting up of the complementation test, you did not want to see a glob, but this time you do. So as it says in the protocol, you can use a toothpick. But what I'm going to do instead is use the pipette tip. So I'm going to use the P1000. I will put a tip on it. I will then open up my HBO yeast plate. I will scoop up some cells with the tip. You are not pipetting these cells up. That likely won't work anyway since it's a solid. You are just scooping so that you now have a small glob at the end of the tip. Then put the tip into the 10 to the 7 tube and scrape off your glob to release it from the tip. Do some stirring and mashing before then resuspending this chunk of yeast to get a more homogeneous mixture. To do that, you see that I am pushing up and down on the plunger at the top. So I'm pressing down, that pushes the air out. Then I will suck up the liquid here. I am slowly releasing the plunger to do that. And I'm going to do this many times until the mixture looks more homogeneous. Now that you have prepared the 10 to the 7, you are ready to move on to the 10 to the 6. So grab the P200 pipette now, set it to 100 microliters, put on a 200 microliter pipette tip, close the lid to avoid contamination of your sterile tips. Then transfer 100 microliters of the 10 to the 7 to the 10 to the 6. Mix it up a bit by pipetting up and down. And then eject the tip into the trash. Get a fresh tip, transfer 100 microliters of the 10 to the 6 to the 10 to the 5. Pipette up and down. Eject the tip and so on until you have diluted all the way down to 10 to the 3. You will see that as I do this, I am moving the tubes down. That way I can make sure that I don't put the mix into the wrong tube. A toothpick full, or in our case, a pipette tip full, is about 10 to the 7 cells. So we have now approximately 10 to the 7 cells in this tube per 1,000 microliters. In this tube we have 10 to the 6 and so on. 10 to the 7 cells is 10 million cells. So if you do the math, that's 10,000 cells per microliter in this tube. So if I were plating 100 microliters, that's 1 million cells. That's a lot. If each of those cells grows into a colony, that will be a lot of colonies. That's why we are setting up this dilution series. So that once you take 100 microliters of the 10 to the 3, that's 1,000 cells per 1,000 microliters, so 1 cell per microliter. And if I take 100 microliters of that, that's 100 cells that will grow into 100 colonies. That's reasonable. Now I'm ready to plate. I will demonstrate this with the 10 to the 7. For each tube, you want to have 2 plates. On each, you will spread out 100 microliters. Make sure to label the bottom. First, I will tear open a spreader. Leave the hockey stick portion of the spreader in the packaging. It is sterile, so we want to keep it that way for the moment. Now I will take 100 microliters and I will put it onto the plate. Now I will take my spreader out and spread this out while rotating with my other hand to evenly distribute this culture. Once you are done, put the lid on, but do not invert quite yet. Let's let that sit for 5 to 10 minutes to absorb into the agar before flipping. That's it for now. See you in the lab.